Good morning, guys. I'm Sir Franz, and I'd like to welcome you to our first lesson on drug development and discovery. This will be our lesson outline. So, through the introduction, we will try to answer the following questions: what a drug is, why do we need to develop new drugs, and what is the better term to use, drug invention or drug discovery? First, a drug is any chemical agent that affects living protoplasm. So, whatever that substance is, whatever that element or compound is, if it affects the function of a living system on the cellular level, that is a drug. So, the study of drugs is called pharmacology. And by definition, pharmacology is the study of knowledge that embraces the source, physical and chemical properties, compounding, physiological actions, and that involves absorption and fate, and lastly, the therapeutic uses of drugs. So let's break down this definition. First and foremost, it's a body of knowledge, which means for us, it is a lifelong pursuit of gaining more and more knowledge through research and study. What does this knowledge involve? Source. Saan nanggagaling yung gamot? Physical and chemical properties. So, by knowing the physical and chemical properties of medications, we can predict how they will act in the body, and we can modify these properties so that it is better suited for use to alter the function of the human body. Compounding of these medications, which is the centerfold in pharmacy, physiological actions, absorption, fate, so what the body does to the drug, metabolism, modification of the structure by chemical processes, and eventually elimination of the drug and therapeutic uses of drugs. So, lastly, we are trying to know how these drugs are better used to treat diseases. Why do we need to develop new drugs? First, because we get sick. And personally, if you will recall an experience of getting sick, that is a feeling that you do not want. And so it is innate in us to find remedies for this sickness. Therefore, it is a natural extension of human behavior to find these drugs and scientifically to modify and improve these drugs, to develop these drugs. And on top of that, we develop new drugs because we have the desire to improve our physiology. So even if we are not sick, we would like our functioning to be higher, to be better than normal. So, for example, if you are already acquainted with taking coffee to study, uh, socializing, you will observe that the function of learning, the function of focus, these are executive functions, they become better if you take in coffee. Without coffee, you are normally functioning. You're at the baseline. But if you take in coffee, and particularly the caffeine inside coffee, you will observe that your focus, your learning, your attention, and ability to calculate improves. So that is the improvement of what is at the baseline, the improvement of our basal physiology. So that's another reason to develop new drugs. Next, we have to ask the question, what is a better term to use? Drug discovery or drug invention? If we use the term discovery, then there is that connotation of chance, of serendipity, that we encounter a drug by way of luck. But if we use the word invention, then that connotes a deliberate and scientific process or approach into knowing these drugs, discovering these drugs, and eventually modifying and improving these drugs. So for me, if I were to choose what the better term is, drug invention or drug discovery, I would go with the term drug invention because that is what we do in pharmacy. We approach it in a scientific, systematic way. So it's either drug invention or drug development. Let's just go through the medicines throughout the ages, the human fascination and infatuation, which means we really get in love with medicines, with chemicals that alter biology, is ancient. Ibig sabihin, noon pa lang, yun na yung relationship natin sa mga gamot. And it results from long experience and dependence on plants. So you will observe that much of the drugs that we use right now, the aboriginal, the prototypical drugs that we have and we use to treat diseases, find their origin in plants. And so through this course that we will be having, drug invention and drug development, we will be trying to get a grasp of how we can utilize these natural chemicals and try to convert them into medicines and drugs. So, in the past millennia, which means in the past thousand of years, we have learned about coffee and caffeine, how it is a central nervous system stimulant. And right now, upon further research, we are discovering that intake of coffee decreases all-cause mortality by 5% and it slows down neurodegenerative processes. For example, Parkinson's and Alzheimer's. Belladonna, you have here a picture of dilated eyes. It was used by ballerinas around 1700s, 1800s because it would cause these women to be more beautiful because of pupillary dilation or mydriasis. And that is due to the effects of atropine, Atropa belladonna, on the iris and muscles that control pupillary dilation. So that they would have bigger pupils. And that will be perceived as something more beautiful. Mawang is a Chinese drug, but the active ingredient there would be ephedrine. Ephedrine increases the activity of the sympathetic nervous system by increasing the release of catecholamines and by preventing the breakdown of catecholamines by way of monoamine oxidase. So tumataas yung levels ng epinephrine, and that presents as a cardiotonic or a vasopressor effect. So it increases blood pressure and it helps patients who are in shock. Curare, you have here the blue frog, is actually a poison. So the American Indians would coat the tips of arrows with the back and mucoid substance on the back of these frogs, and this will serve as a poison so that when the arrow hits the target, that prey is immobilized, because curare contains tubocurarine, which is a non-depolarizing neuromuscular blocker. So it binds to the acetylcholine receptor but prevents action of the acetylcholine receptor. So without acetylcholine activity, you won't have the end motor plate activity. So walang muscular contraction. You have opium, and that's the opium poppy plant. And opium, the active ingredient would be morphine, that causes you to sleep, but at the same time, that provides potent analgesic activity. So thousands of years ago, they know that already. And you have poisonous and psychedelic mushrooms, which elevate the state of mind, but at the same time can be used as poisons. For the past centuries, which means hundreds of years, we have discovered that dyes have potent biologic activity. They can be used as anti-cancer medications, so they have cytotoxic effects. And dyes can also be used as antimicrobial medications. So for example, you have the dye Salvarsan. You have here a dye also, Prontosil, and that is the prototype for an antibacterial agent in the form of sulfanilamide. So that is um, it's a protein synthesis inhibitor. Okay. So what is the usual process? We have substances from nature, and these are biological substances, and we apply them into the human body. So drug molecules, with the exception of a few naturally occurring hormones, most drugs are small organic molecules. So usually they will have an atomic weight of less than 500 daltons. So ibig sabihin, yung mga gamot natin, majority of them would be small molecules. Exceptions here would be peptides and proteins that are derived from living cells. Protein therapeutics were uncommon before the advent of recombinant DNA technology, where we get genes that eventually are translated into proteins and insert these genes into E. coli or yeast, and have the E. coli or yeast, by their own central dogma and translation, synthesize the proteins of interest for us. And we get the proteins and purify them and inject them into the human body. So you have here, for example, porcine and bovine insulin. So ibig sabihin yung pancreas noong pig and cow, we homogenize it and we isolate from it insulin. And the insulin we inject into the human, and eventually, because we share relatively large sequences of amino acids, yung human insulin and porcine bovine insulin, similar, it will also be able to produce effects in the human body. Okay? And that effect would be a decrement in blood sugar. So what is our usual approach to drug invention? We screen a library of chemicals. So, sobrang daming chemicals. And then, um, we already have at the back of our mind which chemical structures potentially produce a pharmacologic effect of interest. So we have lots of chemicals, we screen a library, and then after screening, there are some of those chemicals that have a higher chance of producing a certain pharmacologic effect. And these are what we call hits. Hits are identified in the screen, and we modify the hits by way of synthesizing derivatives, by defining the structure activity relationships, and we modify those structures, optimizing their parameters, and we transform them into leads. So you have hits, and then they undergo synthesis, defining of the SARs, and optimization, and then they are transformed into leads. And eventually, the leads, step number 3, are incorporated into a drug delivery system. And ito na yung ibibigay natin sa um, animals, preclinical trials, or to humans, clinical trials. So alternatives, um, we synthesize and focus on close chemical relatives of a substance known to participate in a biological reaction of interest. So for example, an alternative would be meron ka ng known substance that has a known biologic effect, and from there, you just take that substance and tweak it, and you have a new drug. So that's an alternative approach to drug invention. Next, we have targets of drug action. It is not enough that we are able to identify substances that have particular effects in the body, but we also have to know where that substance acts. So these are targets of drug action. What are crucial questions to ask? Number 1, can one find a drug that will have the desired effect against its target? Number 2, does modulation of the target protein affect the course of the disease? So first, we identify a target. Second, if we modify the activity of that drug target, will it affect the course of the illness or the course of the disease? Will it have a beneficial effect on how that patient has the disease, so the experience of the disease, or does it have a beneficial effect on mortality? Will it decrease the mortality and increase survival? Does the project make sense economically? Because you will understand that development of drugs is a multi-million dollar process. So kung ipupursue ba natin itong um, drug development pathway na ito, will it be economical? Will the costs be low and the returns high? Kasi hindi pwedeng lugi. Okay? So, for example, we have here insulin as our previous example kanina. The picture on top would be the usual phenotype of a patient with diabetes mellitus. So they don't have insulin activity. And remember that insulin lowers blood sugar, but at the same time, insulin is the most important anabolic hormone. It is the hormone that allows your body to build muscle, to synthesize proteins and to build fat, to store fatty acids in the form of triglycerides. So if you have type 1 diabetes mellitus, for example, where your beta cells have been destroyed by the immune system and you are not able to produce insulin, you will have that body. No muscle, no fat, high blood sugar. So here in the image, a schematic representation, you will see how insulin functions. So insulin binds to the insulin receptor, and you understand that the insulin receptor has subunits, an alpha subunit that interacts with insulin, and the beta subunit, which is a transmembrane protein that produces the effects of insulin inside the cell. So there will be signal transduction, a lot of phosphorylations of proteins, but at the end of the day, it will result to cellular growth, protein synthesis, storage of glucose in the form of glycogen, and glucose transport. It upregulates the vesicles that contain GLUT4 and allows these GLUT4, glucose transporters, to be inserted into the cell membrane so that glucose is able to enter the cellular space from the interstitial fluid. So that allows glucose to enter the cell and eventually leads to a decrease in plasma glucose. So that's how glucose happens. Ngayon, can you just imagine how they developed insulin years and years ago? So that is through the wisdom of Dr. Banting, who discovered insulin. So let's try to situate ourselves in the mind and in the shoes of Dr. Banting. Can I find a drug that will have the desired effect against the target? So insulin, can I find insulin? And where will I find insulin? And where will insulin act on the insulin receptor? Does modulation of the activity of the insulin receptor affect the course of the disease, which is diabetes mellitus? Years ago, type 1 diabetes mellitus, before the discovery of insulin, is a fatal disease. And lastly, how will I modify the production of insulin, injectable insulin, from, for example, cadavers, dead people, getting the pancreas of dead people, or getting the pancreas of pork and cows, into something that is more economical, into something that is more sanitary. Okay? So, yun yung question number 3. Lastly, I touched on this a while ago, that in the development of drugs, we have to first investigate its effects on biological models in animals. So that is a preclinical trial. And once we use the term clinical, we are already referring to administration of drugs to humans. So this last slide is very important, and I know it paulit-ulit na tong dinidiscuss sa inyo, clinical trial phases. The stepwise manner by which we test drugs in humans. Stage 1 or phase 1. First, in humans. We utilize 10 to 100 participants, and these participants are usually healthy volunteers. And occasionally, if we are dealing with a disease that is very rare, stage 1, patients with advanced or rare disease. We test here the safety and tolerability of the drug. It lasts up to a year, and the estimate would be about $10 million for phase 1, and you have a 50% success rate. From that 50%, you can proceed to stage 2 or phase 2. This is the first time we give the drug in patients, which means these are already participants who have the disease. So we employ up to 500 participants, and these are patients and these are subjects that receive an experimental drug. The goal of phase 2 would be to determine if the drug is efficacious and to determine what the appropriate dose is. So this usually lasts for 2 years, and we usually utilize up to 20 million US dollars, and this has a success rate of 30%. Now, phase 3, if you think about it, would be an expansion of phase 2. So we have a multi-site trial. Ibig sabihin, at the same period of time, sobrang daming bansa or sobrang daming ospital ang nagko-conduct ng trial. We have up to a few thousand participants, and same sa phase 2. Our subjects are patients with the disease receiving an experimental therapy. So our goal here would be to confirm the efficacy of the drug in a larger population. And this lasts for up to 5 years. Ito sobrang mahal na. 50 million US dollars with an estimated success rate of 50%. Now, after the drug passes through phase 3, the drug enters the market where it is available for purchase. So this is already your post-marketing surveillance. And kung sino makakabili, they will be the ones exposed to the drug. So these are new drugs available in the market. So our subjects here would be patients in whom the drug or treatment is indicated. And the goal would be to determine adverse events that will present later. Drug-drug interactions and compliance. And it has no fixed duration. So we have phase 1, 2, 3, and 4. So that will be the end of this lecture. We have discussed drugs, historical view of drugs, how we develop new drugs, how we try to identify targets of drug action, and lastly, clinical trial phases. Thank you for listening.